Hello everyone, welcome to GTC. In this video, we are going to discuss about growth phase of bacteria. So bacteria, we basically culture this bacteria in a culture medium. When we place this bacteria in a culture medium, they basically show different type of growth activity. Let me explain you by this graph. So for suppose, let's simply assume I'm telling a story. Let's assume that. For suppose you are you are going to stay in a hostel. Okay, so what is the scenario of first month of hostel? So in first month of hostel staying, so okay, simple you this scenario you assume in first month of hostel staying, what happens? So some people have make friends, some will not make friends. Basically, you are going to adapt to that hostel environment. Okay, you are going to adapt to that hostel environment. Some people might leave the hostel because they're not able to adapt that to that hostel environment. Okay, after 1 month, so when you completely adapted to the hostel environment, now your friendship will be more. You will go for late night talks, late night parties, late night studies, all these things. So this called as once you acquainted, your frequency will be high. Okay, you assume your frequency will be high because now you made the friends. Now you are enjoying your hostel life. Okay, so once you peak reach to that enjoyment, for suppose exam has come. Now what happens? Now there will be separation of friendship, breaking of friendship and all. So that high enjoyment time will start decreasing. So we can write frequency decrease. Okay. So this is exactly what happens to the bacteria when we introduce bacteria with a culture medium. So the first step is called as lag phase. So lag phase is the phase when bacteria bacteria are inoculated with new culture medium. Okay. Bacteria are inoculated with new culture media. So initial phase here, bacteria are just acquainting with the culture media. Here, what happens? Some bacteria might not acquainted with that culture medium or not compatible with that culture medium. So that's why death of some of the bacteria also takes place. So this phase is called as lag phase, or we can call it as a initiation phase. Okay. After that, as I told you, when the friendship uh, make and the enjoyment is started, your frequency will be high. So in log phase, the growth frequency is very high. In the log phase, growth frequency is very high. Here reproduction is at maximum peak, so bacteria start doing binary fission for their reproduction. So the reproduction phase is very high, so that bacteria number basically doubles at that time. After that, stationary phase, or also called as plateau phase. So here growth is equal to death. Okay, in this page, growth is equal to death. And the last phase is death phase or decline phase when death is more. Okay, death is more as comparison to growth, that is decline phase. So question can be asked, why this death occurs in bacteria? So this thing will be discussed in this tabular form. So first page is your lag phase. So lag phase basically the period between inoculation and beginning of the multiplication. The first step is basically inoculation and beginning of the multiplication. Okay, so you have inoculated this bacteria and the bacteria basically start multiplying. Next is the coenzymes and other essential molecules are synthesized by bacteria cell during this stage. Bacteria will start synthesize enzyme and reproduce the enzymes, coenzymes. That means this bacteria basically prepare itself for the cell division. Okay, next is log phase, also known as exponential phase. Here, reproduction and growth is at highest rate. As I told you, frequency is high. Number of bacteria basically doubles with each generation. Next is stationary phase. So, reproduction and death rate equalize, and hence this bacteria population attains this plateau. As I told you, reproduction and death basically equalize. This phase basically nutrients become scarce. Now Bacteria started utilizing this nutrient. You have not added any fresh nutrient. That's why nutrient level started decreasing. Okay. So waste products, and main thing is waste product accumulation. Waste product accumulation, or we can call toxic metabolite accumulation. So when bacteria metabolize, so toxic metabolites are formed that basically accumulated, and that toxic metabolite can affect the death of other bacteria. So that happens in basically this decline phase. So decline phase basically bacterial death due to a lack of nutrition and temperature and environment. Death or decline phase is also known as logarithmic death phase. I hope you understood. So let me summarize. So there are three, four phases that occur during bacterial growth. So initial phase is called as lag phase. This is basically the inoculation of the culture media to the bacteria. Okay. In this phase, bacteria started synthesizing their enzymes, coenzyme, and other things for their development. Next phase is called as log phase, also known as exponential phase. Here, growth and reproduction is at highest peak. Next is a stationary phase. Here, growth is equal to death, and here toxic metabolite have started accumulating. When the toxic metabolite concentration is more bacteria will die. So, question can be asked: what is the cause of death phase? One is your toxic metabolite accumulation. Second is deprivation of nutrients, deprivation of nutrients. So you can add this fresh nutrient. If you add fresh nutrient, there might be a chance that bacteria will start reproduce. Okay, so I hope all of you understood what we discussed right now. So for more such content, follow our YouTube channel GDC Classes English. Stay connected with us for more such content. Thank you so much.